Welcome to this lecture on parasitology. Today, we are going to explore the fascinating and sometimes unsettling world of parasitic nematodes. Imagine your body is a luxury hotel, and suddenly, uninvited guests arrive who not only refuse to leave, but also eat your food right off your plate. These microscopic invaders affect billions of lives and cause massive agricultural losses worldwide. By the end of this session, you will understand not just how these creatures survive and thrive, but also how scientists and doctors use mathematics to track and fight them. Let us dive in. To begin, let us understand what these uninvited guests actually are. Parasitic nematodes are commonly known as roundworms because of their cylindrical, spaghetti-like shape. But why should we care about them? Because they are incredibly successful survivors that have mastered the art of invasion. They can sneak into a host organism through contaminated food, dirty drinking water, or even by burrowing directly through the bare skin of your feet when you walk outside. Once they make it inside, they set up camp in nutrient-rich areas, like the intestines or the lymphatic system. There, they quietly steal your nutrients, causing a lot of hidden damage over time without immediately killing the host. Now that we know how they enter the body, let us look at how they survive such hostile environments. Biologically, parasitic nematodes are unsegmented, cylindrical worms belonging to the phylum Nematoda. Unlike their free-living cousins that harmlessly inhabit soil or water, these parasites strictly rely on a host organism to survive, grow, and reproduce. The human stomach is filled with highly corrosive acids designed to break down food and kill pathogens. So how do nematodes survive being swallowed? They have evolved a highly specialized anatomical structure called a cuticle. This tough, impermeable outer layer acts like a biological suit of armor, perfectly protecting them from being digested by the host's harsh stomach acids. Beyond their physical armor, these parasites also have incredibly sophisticated ways of reproducing. Parasitic nematodes undergo complex life cycles that often involve multiple distinct developmental stages. They start as microscopic eggs, hatch into several active larval stages, and finally mature into reproductive adults capable of producing thousands of new eggs. Some of the most common examples that affect humans include the giant roundworm, hookworms, and filarial worms. What makes these specific nematodes so dangerous is that they have adapted perfectly over millions of years to evade the human immune system. They can live inside a person for years, constantly reproducing and continuing their life cycle, right under the radar of our body's natural defenses. Understanding their biology is fascinating, but it is also a matter of global survival. Studying parasitic nematodes is absolutely critical for maintaining global public health and ensuring agricultural food security. In human populations, these worms are devastating. They cause severe malnutrition, stunted physical and cognitive growth in children, and debilitating chronic diseases like elephantiasis or severe anemia. But humans are not the only victims. In the agricultural sector, plant parasitic roundworms, like the root knot nematode, attack plant roots and destroy billions of dollars worth of essential crops every single year. If we did not deeply understand how these parasites reproduce and spread, we would have no way to develop effective deworming medications or design proper sanitation interventions to protect vulnerable communities. Because these parasites cause such massive damage, we cannot just know they exist. We must actively track them. To fight parasitic nematodes effectively, doctors and epidemiologists must measure exactly how widespread and how severe the infections are within a community. This is where biology meets mathematics. Parasitologists use specific mathematical formulas to quantify the burden of disease. By quantifying exactly how many worms are in a population, medical treatments can be targeted exactly where they are needed most. The two most important metrics we use are the prevalence rate, which tells us how widespread the parasite is across a population, and the intensity of infection, which tells us how many worms are actively living inside an individual host. Let us look at the first of these crucial metrics, the prevalence rate, and where its formula comes from. Conceptually, prevalence is just the fraction of a group that is sick, converted into a percentage so it is easy to understand and compare. The formula is P equals I divided by N, all multiplied by 100. Here, P represents the prevalence rate expressed as a percentage. The variable I stands for the total number of infected individuals who tested positive for the nematode. The variable N represents the total number of people in the population being tested. Think about how this behaves physically. If the number of infected people, I, increases while the total population, N, stays the same, the prevalence, P, will increase. This tells health workers that the parasite is spreading more widely through the community. Now that we have our formula, let us see this with a concrete example. A global health team visits a rural village to test the population for the giant roundworm. We are given that the village has a total population of 800 people, and stool samples reveal that 320 people are actively infected. We are asked to calculate the prevalence rate. We choose the prevalence formula because we need to find the percentage of the whole group that is sick. First, we substitute our known values. 320 goes in the numerator for I, and 800 goes in the denominator for N. We divide 320 by 800, which gives us a decimal value of 0.40. Finally, we multiply by 100 to convert this raw fraction into a percentage. The final answer is 40%. Physically, this means out of every 100 people, 40 are hosting parasites, indicating a highly endemic area requiring immediate mass deworming. We have successfully calculated how widespread the disease is, but that brings us to an important question. How sick is each individual person? Prevalence only tells us if someone is infected, not how severely. To measure the intensity of intestinal nematodes, we have to look at the parasite's reproductive output. We do this by counting the number of microscopic eggs passed in the host's feces. This standard measure is known as eggs per gram. It is a direct proxy for the number of adult worms living inside the patient. If a doctor sees a high egg count, it tells them that the patient has a much heavier, more dangerous worm burden inside their body, which could lead to severe complications like intestinal bleeding. To find this intensity, we use a specific formula. Conceptually, because solid waste is hard to examine, laboratories dilute it into a liquid suspension. This formula scales the egg count from that tiny liquid drop back up to represent the original solid sample. The formula is E equals C divided by V, all multiplied by F. Here, E represents the eggs per gram. The variable C is the raw count of nematode eggs actually seen under the microscope. The variable V represents the volume of the liquid suspension placed on the slide, usually measured in milliliters. Finally, F is the dilution factor, accounting for how much water was mixed in. Physically, If the raw egg count C increases while the volume and dilution stay the same, the value of E increases proportionally, indicating a much more severe infection. Let us apply this formula to a clinical scenario. A laboratory technician is examining a patient suspected of having a severe hookworm infection. We are given a dilution factor of 50, a slide volume of 0.15 milliliters, and a raw egg count of 120. We are asked to calculate the total eggs per gram. We use the intensity formula to find the true concentration in the solid waste. First, we substitute our values: 120 for C, 0.15 for V, and 50 for F. We divide the raw count of 120 by the volume of 0.15. This gives us 800, which is the concentration in the liquid. We then multiply this intermediate value by the dilution factor of 50 to scale it back up. The final answer is 40,000 eggs per gram. Physically, this massive number indicates a life-threatening infection requiring immediate, aggressive medical treatment. Now that we have calculated both metrics, let us look at a diagram that visualizes how they connect. On the left side of the screen, you see a population of human figures. Some are colored red, representing infected individuals, while the gray figures are uninfected. This entire group represents our population, N, and the red figures represent I. The arrow shows how this population data feeds directly into the prevalence rate formula, giving us a macro-level view of the disease. On the right side, we zoom in on a single infected individual. You see a circular microscopic view of a counting grid containing brown, oval-shaped nematode eggs. The labels clearly point out the raw egg count, C, and the liquid volume, V. This microscopic view feeds into the eggs per gram formula below it. This diagram beautifully illustrates how parasitology requires both a wide lens to see the community and a microscopic lens to treat the individual. We have covered a lot of ground today, so let us summarize the most important ideas. First, parasitic nematodes are highly adapted, unsegmented roundworms that cause immense suffering and economic loss worldwide. They survive using specialized structures like their protective cuticle and complex life cycles. Second, we learned that fighting them requires mathematics. The prevalence rate is our tool for measuring how widespread the infection is across a whole community, helping us target public health interventions. Third, the eggs per gram metric allows us to measure the exact intensity of the infection inside a single patient, guiding clinical treatment. By understanding both the biological nature of these parasites and the mathematical tools used to track them, we can develop strategies to interrupt their transmission. You now have a solid foundation in the epidemiology of parasitic nematodes, which is the first step in eradicating these hidden invaders.